We're going to take the challenge of moving to our first speaker. I will take the opportunity to introduce Dr. Jyoti Nangalia. She has been with us before. She's a clinician, scientist, and consultant hematologist at the Sanger Institute in the United Kingdom. And she utilizes sequencing technologies to help understand why blood cancers originate and evolve and how this relates to normal development and aging. And she will talk with us today about MPN molecular biology. Thank you.
Dr. Nangalia, can you accept the invite?
Hello. I can't hear you guys now. 1 second.
Yes, we can hear you.
Yeah, lovely. Hello, everyone. Good morning there. Good afternoon here. Now, let me see how I screen share. Share screen.
It's the second button from the right.
Can you see my slides?
Yes, we can.
And you can hear me too?
Yes, we can.
Lovely. Okay, well, I'll get started then. Thank you very much for that lovely introduction. Apologies in advance for two things. Number one, we're stuck at home under a British lockdown here. And so if my internet connection is slightly unstable because the rest of the family seem to be streaming on Netflix, then apologies. Second of all, if I stop them from streaming Netflix, then they'll all come here and there'll be a bit of Zoom bombing during the talk. So one or the other may happen. Apologies in advance. So today I'd like to take you through some exciting research that we have been doing over the last months, but I'll start with a bit of an introduction to what causes MPNs first. So I'm fascinated by hematopoiesis or blood production, and in the top left corner you can see a picture of what blood looks like underneath the microscope, with the red cells that carry our oxygen, these slightly larger cells, 3 of them that are different types of white cells fighting bacteria and viruses, and the very small purple blobs that you can see, the ones that help our blood clot, those are the platelets. And within the bone marrow, which is where blood is made, it's a much richer picture, which is what the background picture is. You have the progenitors and the parents of all these mature cells that we see in the peripheral blood. And what we're most interested in is these stem cells that make all of our blood and the progenitors. Because in the myeloproliferative neoplasms, what happens is that these blood stem cells and progenitors are overactive. So much so that one produces too many of mature blood cells. Oh, it says here, are the slides frozen? Can everyone see my slides?
Yes, we see your the slide with the blood stem cell.
Lovely. Okay, so it might just be at the user end. Okay, so in myeloproliferative neoplasms, what happens is the blood stem cells and the progenitors are overactive, and that leads to an overproduction of most commonly platelets and red cells. And there are 3 things that I want to talk about today: what causes MPNs, and then these 2 questions that I get asked all the time in clinic, and I'm sure many of the clinicians here today will echo that sentiment, and patients really want to know, how long have I had this for? And how fast did it grow? Is it still growing? And we now have answers to all of those, really. So first of all, what causes it? Now, on the left-hand side is a pretty exciting picture. It's the very first time in history that the number of chromosomes within our cells was counted. Up until then, we knew how many chromosomes apes had, but we didn't know how many chromosomes humans had. And this is the first picture of that. And within our chromosomes, as we unravel the chromosome, what we see at the bottom here is the code for life. It's our DNA, and every single one of our trillions and trillions of cells in our body has to carry around with it the DNA for the code for life. Now, what happens within this DNA is that about 2% of the DNA codes for the proteins that we use within our body. And one of those proteins is JAK2. Now, JAK2 is a signaling protein. It sits within our cells, and it passes messages from the outside of the cell to the inside of the cell to tell a stem cell what to become. Should I become a red cell, a platelet, or a white cell? And what happens in MPNs is that there's a particular part of the gene that gets mutated, and it's the part of the gene that switches the JAK2 protein off. And when that part of the gene gets mutated, that off button stops working. And in effect, what happens is you end up having a permanently on JAK2 protein. So what then happens is this is a stem cell, and the stem cell hasn't quite decided what type of mature blood cell it's going to be. And EPO is a hormone that we make near our kidneys that tells our stem cells to make more red cells. And JAK2 is normally the protein that shuttles between EPO and the cell to tell it that, there's EPO around, we need to make more red cells. But when JAK2 is permanently on, irrespective of how much EPO an individual has, the bone marrow is continually making more red cells, or it may be more platelets, whatever it is. So essentially we end up with a situation where with the JAK2 mutation, you end up having too many red cells. And the discovery of the JAK2 mutation back in 2005 actually revolutionized medicine, because it changed the way we diagnose patients with MPNs. Now, even family doctors can test for a JAK2 mutation and know instantly whether a patient has underlying polycythemia vera, which is characterized by too many red cells, or indeed essential thrombocythemia, where patients have too many platelets, or myelofibrosis, an advanced form of the condition. If you have a JAK2 mutation, then it's pretty much diagnostic for this condition, and it's also led to the development of treatments that we often use for our patients now. But, about half of the patients did not have a JAK2 mutation up until a couple of years ago. We didn't really know what drove their disease. And in those patients, they had to undergo a whole host of tests, including bone marrow biopsies. We did find in a few patients that, there were other genes that, when mutated, mimicked the consequences of JAK2, but they weren't present in very many patients. During my PhD in Cambridge with Tony Green, we were studying the genetics of some of these patients that didn't have JAK2 mutations, and we and others uncovered mutations in a gene called calreticulin. And that was really present in pretty much the rest of the patients that did not have a JAK2 mutation. So it really gave a sense of completeness to the field now that for the vast majority of patients, about 90% of patients, you can diagnose their MPN with, very simple genetic tests looking for very specific mutations in a couple of different genes, JAK2, calreticulin, indeed MPL. And because the calreticulin mutation leads to a change in the size of the protein, what you can do actually is, rather than just test for the actual mutation, you can look to see whether a patient has a normal sized CalR gene, or a slightly bigger, a slightly more smaller size. And if they do, then that's in keeping with it being a mutation. So in effect, changes in the JAK2, the CalR, and MPL, which I haven't yet mentioned, but it essentially does the same thing as a CalR mutation. They drive MPNs because they become mutated and they signal to our stem cells in our bone marrow to make too many mature blood cells. And 90% of patients have one of these mutations. But about 60% of patients have mutations in additional genes as well. Often many of these mutations are found in the healthy population as one gets older. And so a key question in the field has been, well, what is the difference then between those that have these mutations and have no problems from them versus those that get the actual disease, the MPN, and have problems from it? And that really relates back to the questions that patients ask us, which is, how long have I had it for, and how fast has it been growing? And that's what I'm going to go on to next. How long have I had it for? Now, in the field, I think if you ask patients and if you ask doctors, how long do they think they had their MPNs for? Sometimes patients report, well, I've not been feeling very well for a couple of years, and perhaps it's been going on. Sometimes it was just picked up by accident because of a blood test that was done. And you sometimes wonder, well, perhaps the patient did have it for many years before. But if I had to guess, I'd probably say a couple of years, maybe 5 years before. But we wanted to actually address this question, and we set about doing that. And the way we did that is this is data that isn't published as yet, but is in the process of being so. We took 10 patients aged between 20 and 76 years at diagnosis, so a whole type of different ages of presentation of MPN. Some patients had ET, other patients had PV, some patients had MF. And what we did is we took their blood or their bone marrow and we separated it into single cells. And we then took each single cell and grew each single cell into a colony of cells. And here in this little picture is a little colony of red cells. It just looks like a little pinprick, even to the naked eye, growing in a dish. What we then did is sequence the DNA of that colony, the whole genome. So the genome, the human genome is a couple of billion base pairs, and we sequenced the whole genome within these colonies. So we ended up with whole genome sequencing of about 1,000 colonies. Now, this is quite an unusual thing to do. Normally, when you hear of sequencing, you take the blood and you just sequence it once in a patient to find out what mutations are present. But what we're doing here is sequencing multiple times of single cells within an individual patient. So we kind of sequenced 100 times per patient. And the reason we wanted to do this, which may seem a little bit crazy and expensive, is we wanted to build a family tree of mutations. So just like you might go to a village and you want to see how all the individuals in the village are related, you might then look at each individual and what do they look like, and their features and their address, and who their siblings are and their cousins and their aunts and uncles, and you want to build an entire tree right back to those ancestors that first came to the village. We wanted to do that, but with mutations. And the reason we can do that with mutations is right from when we existed as a single cell, where the egg and the sperm combined into a zygote, every time that cell grows into 2, then 4, then 8, we're acquiring mutations. Not mutations that cause problems, just errors in our DNA sequencing, because we have 2 billion bases within our DNA, just random errors. They don't cause any problems, but they become barcodes, such that at any one snapshot during our life, each of our cells is slightly different from the other cells. We're a bit of a mosaic. Our cells are slightly different to what the DNA was when we were born, because we've been acquiring these individual mutations. But in doing so, we can trace back the ancestry of these cells right back to birth. And so this is what we did with the MPNs. Now, when we did sequencing of the individual colonies, we found many more mutations than we thought to be present when we just sequenced the blood, showing that actually there are many more mutations present in our blood in both patients with MPNs and in healthy individuals than we would otherwise pick up. And then we drew these trees. We built family trees using mutations. And the way we did it is if mutations were present only in one colony, they went on the end of the tree. And if mutations were shared between 2 colonies, say colony A and B, they went in a branch above. And we sort of took all the mutations and drew essentially a family tree. We could then ask on which branch of the tree is JAK2, or which branch of the tree is the other mutation that we know the patient to have. And we could then assign mutations to branches of the tree. And what in effect we're asking is, imagine our blood development as a tree, an actual tree in the garden. We're asking, is the JAK2 mutation on the trunk, or is it on one of those early branches, or is it right at the end of the tree, at the tip of the branch? And that gives us an idea of when it occurred during the lifetime of the development of blood. But we could go one step further than that, because we know that the number of mutations we acquire in our blood stem cells is roughly about 18 to 20 mutations per year. So we can then put time on those branches, and we can say, right, this is how old the patient was when they got their JAK2 mutation. So we did that in our patients, and this is now our family tree for our first patient who was diagnosed with ET at the age of 21. And on the left-hand side, you have normal blood. That's just being produced, stem cells that are dividing over our life, producing blood. And on the right-hand side, you have the blood that's being produced as part of the stem cells that have a JAK2 mutation. And the JAK2 is on a branch that was acquired by the very latest 1.3 years of age, but possibly whilst in utero. And the pattern of branching also tells us how quickly the mutation and that stem cell started to grow, which I'll come on to later. We were really surprised by this because this was about 20 years before the patient was diagnosed. So we went looking ahead, and in this patient diagnosed with PV at the age of 31, their JAK2 mutation also was acquired in childhood at the very latest by 8.5 years of age. And they had other driver mutations that we know to be present in patients with MPNs and indeed healthy individuals, and those too were acquired in childhood, as shown on the left here. Some patients had lots of mutations in their blood, producing blood when we measured them. And in this patient with PV diagnosed in their 30s, the JAK2 mutation was acquired within a few months of birth, but probably in utero and before birth. And indeed, one of their other driver mutations was definitely acquired in utero. In some patients, some of these driver mutations, we didn't find this for JAK2, but we did find this for some other mutations. They were acquired within weeks of life. And what's then happening to some of our patients by the time they present is that their clone has already had a chance to acquire multiple driver mutations, often every 10 years. Sometimes JAK2 was acquired second in 2 of our patients, slightly older patients who presented with ET in their 50s or 70s. The JAK2 occurred as a second event on a background of a driver mutation that had already led to a clonal expansion. So really, that was quite surprising to us, and it gave us a sense that in MPNs, perhaps what's happening is that it isn't suddenly something that happened in the patient's life that resulted in this cancer being caused. It's more something that's been a slow burner and has been growing for a really long time. And I have the sense that this is probably happening in everyone, you know, every human being, but it's just the rates at which those things are growing that determines whether someone is going to get disease or whether those clones are going to remain small and undetectable. And when I've mentioned this to patients before, some of them have been quite surprised, but others have said, actually, you know what, it all makes sense because there really wasn't anything that I did that caused this, and it's probably just part and parcel of how my bone marrow has been developing over life. And I think that's what we think might be happening, but we don't understand it fully yet. Moving on to how fast did it grow, we then went back to those same patients and took multiple time points from their blood to see how the fraction of the MPN was growing in their blood. And we compared that to the pattern of branching in the tree that I explained earlier. The pattern of branching tells us whether something grew quickly or slowly. And we were then able to assign a rate of growth to every single clone historically. So every row here is a patient and a clone. And what we can see in these 3 rows, these are 3 different mutant clones in one patient. One is growing at 10% a year, and one is growing very fast at over 200% a year in the same patient. But also that patients that just had a JAK2 mutation and nothing else, their clones were growing at different rates, suggesting that it's not JAK2 per se on its own that determines that an MPN will be caused. Of course, one does need a JAK2 mutation or whatever else it is that's driving the MPN, but over and above that, it's how fast that grows in that individual patient that also then determines, you know, the rate at which that clone is going to grow and whether that patient's going to develop complications or not. So I believe that when you get a JAK2 mutation, you know, if it's growing really slowly, then there's a chance that it might just die out and not cause any problems, which is what we sort of modeled mathematically on the left. But also the rate at which it's growing actually also determines when a patient might get disease. So in the slowest growing patients, they didn't get their disease until 50 years after they acquired their JAK2 mutation. And in those patients in whom the JAK2 we predicted was growing very fast, they actually presented with their disease within 20 years. So if these JAK2 mutations then occurred well before diagnosis, the immediate question I get asked is, well, could we have detected it? And that depends on the sensitivity of your tests and how low a level of JAK2 a current test can pick up. And I still think that we probably could pick up in these individuals the mutation with really sensitive tests between 10 and 40 years before diagnosis. However, that doesn't mean to say we should be doing that, because I think if we did that, then maybe pretty much lots of people would be positive. So we need to combine that detection with prediction. if we tested everyone, I think we'd find lots of, we know that there's JAK2 mutations present in the healthy population. We anticipate 1 in 100, possibly even more frequent. The key question then is, in which individuals is that clone growing? And I think if we could then predict that, which is the next phase of our work, we could then determine which patients are at risk of possible complications in the future. But this is all work to be done. So in summary, in MPNs, we over the last 10 to 15 years, we really have a sense of understanding of what drives these conditions. And recent work that we've been doing has given us a sense of when these mutations are acquired and how they grow, which is really quite different to what we would have believed. And I believe that in the future, perhaps we could combine this information to try and understand how best to maybe screen populations and predict which patients are at risk. But that, of course, is future work. I'd like to thank our funding bodies and my lab, in particular, Nick Williams, who did much of this analysis. Thank you very much.